Thank you. It's a great pleasure to be here and really enjoyed following in Satellite Bio's footsteps. We are not talking about liver disease. Our target indication is type 1 diabetes. And as you can see on the screen, our technology aims at enabling transplantation of allogeneic cells, in particular islets, whether stem cell or donor-derived, into people with type 1 diabetes. We are a preclinical company located in Hamilton, Ontario, Canada. And the key technological anchor of our approach are our synthetic gel formers that we've developed over a couple of decades and brought into our company a number of years ago. And we use them to combine them with different beta cell payloads to form hydrogel combination therapies, seen on the bottom left, life spheres, life rafts, and life lines. And their key purpose is to enable transplantation of beta cells to correct blood sugar, similar to the Edmonton protocol, but not into the portal vein of the liver and without the need for systemic lifelong immune suppression. So it's a combination therapy. The cells are very important. The hydrogels are as important. And our particular differentiation factor is that we are developing synthetic polymer-based hydrogel network that are covalently cross-linked for persistence and resiliency. We can shape them with 3D printing into high surface area devices that enable very good metabolic exchange and ensure good islet function after transplantation, hopefully in the long term in human patients. They also offer the ability to contain cells which may be useful for certain stem cell-derived beta cells. I skipped ahead. Let me go back. We have a little animation that shows, that illustrates our process. Let me try that again, of forming our gels. I think this will work now. It shows how we combine our gel formers with different cell payloads into initially a viscous solution that we can shape into life spheres, strings, and patches. And these spontaneously covalently cross-link. We can control their permeability using coatings and other approaches. And together they form a platform of geometry-agnostic cell delivery and protection devices. We'll use the string here to illustrate some concepts of these materials. There's no cell escape from the networks, but they are open enough to allow indiffusion of oxygen, glucose, and other nutrients, and of course, very rapid outdiffusion of therapeutic peptides. While at the same time, we control the compositions to the point where we can shed immune components, cellular and molecular based. So altogether, a retrievable solution platform for immune suppression-free delivery of long-term therapeutic cells. I'll show a couple of slides that illustrate the concepts. On the left, you see a life sphere that's being compressed against a vacuum capillary. You see it deform and it jumps back into its original shape. That's important for long-term function in chronically patient people. In the middle, you see what we call a lifeline, extrusion printed. This is a slowed down video with islets into a gelling bath. You can see the cell centering going on in real time. And that approach can be adapted to form a multi-layer life rafts or patches shown on the right-hand side. These are a little bit smaller than what we plan to implant into the patient, but the islets are contained in the linear string sections of that device. And the high internal surface area that's visible in this little video allows vascularization not only from the outside but also through the device, again, hopefully ensuring a rapid metabolic connection as well as good oxygenation. Our approach is built on, as I mentioned, many decades of fundamental polymer designs geared at making hydrogels. In addition, we've also found ways to ensure that the capsules and other devices are formed in ways that have no physical imperfection that could elicit a mechanical immune response. So these are smooth, they're monodisperse. And we control the distribution of our cross-linked hydrogels as shown in the center. We can cross-link throughout the capsule or hydrogel. We can also cross-link predominantly at the surface as may be required. And of course, because we make it, we also control the composition and any post-modifications that we may want to do in order to further mitigate the immune response of the host. That leads to scenarios such as on the right, where we've encapsulated islets that are implanted, explanted after 6 weeks. The capsules are still clear, the islets are intact, and importantly, there's no swelling, no significant swelling, which means the molecular weight cutoff does not suddenly increase during the implant time, helping to keep out cytokines that we don't want to get into the cells. We are also, in addition to primary human and animal donor islets, we are getting ready for stem cell-derived cells. We show here an encapsulation of HIPSCs that are fitted with Pancella's fail-safe suicide trigger. And you can see on the top left how we've encapsulated cell clusters that expand over time to an alarming size, considering the capsule, do not escape even over 90 days in vitro, and we can ablate them by application of the suicide trigger through the capsule, so the capsule does not interfere with the metabolic function or these trigger functions. On the right, you see GFP-positive HIPSCs that we've encapsulated, placed sub-Q into black 6 mice, and explanted after 95 days. The cells clearly are still viable and express GFP. So these systems work with human cells in mice. And I'll show a few more examples. But first, our most ambitious shape factor, we call them life rafts. They are printed, and this is a slowed down video of a basic test run. We can center cells in these. Not only is the core composition proprietary, but also the printing bath was developed at Allarta, and that allows us to make high fiduciary patches. Shown on the right is I believe a 10-layer patch that would contain islet in a transplant scenario. These patches, shown again on the left, are loaded with islets. We've explored different islet densities, about 15 to 80 IEQ, islet equivalents, per centimeter linear string length. As we know in the field, and the pancreatic or islet transplants are perhaps not as critical as the liver transplant in terms of number of cells, but it's still important to strike a balance between getting the largest number of islets into a viable situation, a viable construct, into the smallest patch or other device that might be tolerable by the patient. So we aim for the highest possible islet loading. Shown on the left is 80 IQ per centimeters. These are live dead stains, so these islets are green because they're alive. The image in the center is not a live dead stain. It's a confocal stack with color coding along the z-axis, just to illustrate how the islets are lined up along the string dimension. And again, you don't see the open nature of this network in these images, but on the right is an explant from an immunocompetent pig showing vascularization, the little dark lines percolating up and down through the patch or the life raft. And we think that's promising as we go forward in this technology. 2 slides on readouts in our preclinical state. This slide shows human donor islets that were implanted into STZ black 6 mice. On the top left, you see how the blood sugar drops, stays constant for at least 50 days. The HbA1c, the long-term glucose averaging marker, decreases as you would expect over that time for mice. And the capsules come out clean without foreign body response, and the encapsulated islets are still functioning and have very good insulin production rates. On the right are some of our early experiments with immunocompetent Yorkshire pigs, where we implanted, and we have explored different implant sites. We see C-peptide for at least a week and viable islets coming out, stained green, up to week 3 so far. And histology of the explants shows very few immune cells and none specifically around our capsule. So again, very promising data, and we're pursuing this both with Yorkshires and with mini pigs now. This slide shows some of our initial work with rat islets going into mice, where we saw functionality up to 300 days with clean explants. And on the right, allo rat transplants, so rat islets into STZ diabetic immunocompetent V-star rats. And you can see the STZ-induced high blood sugar, which is then corrected within a day. We're currently at 3 weeks out. We need to go a little bit longer, But so far, the data are very promising, and we believe these can be key IND-enabling model experiments. Again, at the bottom, we have 2 weeks and we have 6 weeks explants of clean capsules with highly functioning rat islets. So we are at about 6 weeks and no reason why it will not go longer. As we've heard before, and again in the last talk, cell therapies do better when they actually reach the patient. And so we also embarked on a program to develop logistical supports for cell therapies in different formats. One is shown here. It's a system of micro hydrogels that are designed to pack into a jammed gel into which we can embed cells and cell clusters. They have cryoprotective properties. And so when they eliminate free water, we can freeze them down to -80 in liquid nitrogen, and we find that individual cells come out with a very high viability. We've gone up to 90%, so 95%. So that allows us to decrease DMSO level from the standard 10% down to between 0 and 2%. And we are now working to apply the same to cell clusters, which is, of course, a little bit more challenging for diffusion reasons. Here are some early data that speak to just the advantages of such a material for storage of cell clusters. Shown on the left are donor islets and iPSC clusters stored in 3D in a jammed gel of our hydrogels. These gels are fully transparent. There are no scattering systems. And we can prevent sedimentation and aggregation of islets. And we are using that in our research work right now. And we expect that this will have clinical application at some point in the future as well. So I will conclude by summarizing that hope I've been able to show you how we combine highly designed proprietary polymeric gel formers in different forms into combination therapies with different cell payloads. Type 1 diabetes is our initial proof of concept. We are aware of a number of other indications, and this is also a call to the audience to please talk to us if you think your cells may need alloprotection or other delivery forms. Our materials are shape agnostic. The cross-linked networks allow us to build self-supporting high surface area devices. We are now engaged in talks about scale-up and scale-out and manufacturing at a GMP level. We are aiming for logistical solutions to make sure this does not need to be a one-at-a-time product, but can be preserved, whether at 4 degrees, at 37, or under cryogenic conditions. And we have a clear path to the clinic, as validated by some very senior islet transplant surgeons that are very interested in working with us because the patients are working, and both the donor islets and stem cell-derived beta cells are available. We have a very supportive board of directors that are also our investors, and a strong scientific advisory board that have walked some of this path before and are supporting us and keeping our toes to the fire where needed. So with that, I'll conclude. I want to thank you. I want to thank the organizers for this opportunity, and I'm happy to take any questions now or later. Thank you.